Hello and welcome to this next lesson in our topic of homeostasis. In previous lessons, we've looked at the principles of homeostasis, we've looked at body temperature regulation in endotherms, body regulation in ectotherms, and today we need to look at excretion, homeostasis, and the liver. By the end of the lesson, you should be able to tell me what is meant by the word excretion. You should be able to describe the structure of the mammalian liver and explain its functions, and you should be able to identify some structures of the liver from stained images. So, let's start with a keyword. Remember, all keywords are in green. We're going to start by defining the word excretion. Excretion. Now, when you were at primary school, you would have done Mrs. Gren to identify all living things, and the E in Mrs. Gren stands for excretion, and you would have learned that excretion is where we get rid of waste products. So we have a wee and a poo to get rid of waste. Plants get rid of their leaves in the winter to get rid of waste, and also there may be like arachnids and certain insects that lose their skin. When they grow new skin, this would be a form of waste. However, at A-level biology, we need to define excretion as the removal of metabolic waste from the body such as carbon dioxide and urea. It's the worst A ever, such as carbon dioxide and urea. Okay. Now, metabolic waste is a term used to define waste products produced in your metabolism. Your metabolism is all of the reactions taking place within your cells. So metabolism is all reactions taking place inside the cells of an organism. Okay? Now, these would include respiration. Respiration would be part of your metabolism, and in fact, it makes up the majority of your metabolism. We can now define what metabolic waste is. So, metabolic waste. Metabolic waste can be defined as any substance produced in excess by cells and is not used. Now, almost all cells inside your body carry out aerobic respiration, and when they carry out aerobic respiration, they produce carbon dioxide. We don't use carbon dioxide in our body, which therefore is defined as a form of metabolic waste. So we can put that almost all cells in our body carry out aerobic respiration and produce carbon dioxide as waste. Carbon dioxide as waste. Okay. Now remember, in animals, carbon dioxide would be classed as waste because we don't use it for anything. However, inside plants, it wouldn't be waste because plants can use carbon dioxide in the presence of light to carry out photosynthesis. Another example of metabolic waste would be nitrogenous waste produced in the liver. So we can say that the liver produces nitrogenous waste such as urea in deamination or through deamination. Through deamination. The final example of metabolic waste that we can just mention, and what colour haven't I used? We'll go to this one, is various salts are considered as metabolic waste in your body. So things such as chlorides, phosphates and sulfates are considered metabolic waste. So salts such as chlorides, phosphates and sulfates are all metabolic waste. So we've said that excretion is the removal of metabolic waste from the body, such as carbon dioxide and urea. Metabolism is all of the reactions taking place inside the cells of an organism. This includes respiration, which most cells carry out. Any metabolic waste is any substance produced in excess by cells that is not used by the organism. Almost all cells in our body carry out aerobic respiration and produce carbon dioxide as waste, and the liver produces nitrogenous waste such as urea through deamination. Salts such as chlorides, phosphates and sulfates are all considered metabolic waste. But the question is, how does your body get rid of this waste? And this lesson we're going to focus on these two, because I'm going to assume, particularly this one, it might as well be in a foreign language because you don't understand what urea might be and you might not know what nitrogenous waste is, I seriously doubt you know what deamination is. We're going to talk about that today. And we're also going to talk about how your body gets rid of carbon dioxide. So if we can clear the screen, please bear in mind, to help if I did this, please bear in mind this is a this is a recording, so you can pause it whenever you like. I have so many people contacting me going, sir, you go too fast on the video. Yeah, well, you can pause the video whenever you want. So, can we put the subheading, please? Carbon dioxide metabolism. So, carbon dioxide metabolism. So we've said that most cells in your body carry out respiration, and carbon dioxide is some waste made in that reaction. So we can say that most, I'm putting it in red because it's important, cells carry out aerobic respiration and release carbon dioxide into the blood as metabolic waste. Release carbon dioxide into the blood as metabolic waste. Okay, so when cells carry out aerobic respiration, they produce carbon dioxide, which is released into your blood as metabolic waste. Okay, now carbon dioxide released into the blood is carried by either carbaminohemoglobin or hemoglobinic acid to the lungs where it's gotten rid of. So we can say that carbon dioxide is carried in the blood by carbaminohemoglobin Hemoglobin, carbaminohemoglobin, and hemoglobinic acid, in the blood to the lungs. To the lungs where it is exhaled out. Okay, so essentially cells in your body carry out respiration producing carbon dioxide. That carbon dioxide is put into your blood. That carbon dioxide is carried in the blood by carbaminohemoglobin and hemoglobinic acid in the blood to the lungs where it is exhaled out. Okay, now we need to think now about how this is produced, what these two mechanisms are. So we're going to start with carbon dioxide in the top corner, CO2, which is our carbon dioxide. Okay, don't know what's going on with my handwriting today. Carbon dioxide. Now, nice easy one out of the way. We can combine carbon dioxide with hemoglobin to form carbaminohemoglobin. So carbaminohemoglobin. Okay, so CO2 is combined with hemoglobin. That's a nice simple one. So CO2 is combined with hemoglobin. Okay, so CO2 in the blood can be combined with hemoglobin to form carbaminohemoglobin. Other than being combined with hemoglobin, CO2 or carbon dioxide can be combined with water to form something known as carbonic acid or H2CO3. This is known as carbonic acid. Now, in order to become carbonic acid, the CO2, so carbon dioxide, is joined with water by the enzyme carbonic anhydrase. Carbonic anhydrase. So carbon dioxide can be joined with water using the enzyme carbonic anhydrase to form carbonic acid. Now this carbonic acid dissociates in the blood. Okay, and what we mean by that is that it breaks down in the blood. So this dissociates in the blood. So breaks down in the blood to form hydrogen ions. In fact, we'll stick with that colour. So it forms hydrogen ions and bicarbonate ions, which will be HCO3-. So we've got hydrogen ions and we've got bicarbonate ions. Now, hydrogen ions can lower blood pH. And if they lower blood pH, they can cause something known as acidosis. Okay? So, I'm going to put in orange that hydrogen ions in particular lower blood pH, which can cause acidosis. Now, acidosis, I'm going to try and fit it down here, hopefully we can, is a key word. Acidosis is an illness caused by low blood pH. So, illness caused by low blood pH, and it has the symptoms of drowsiness, breathlessness, and a rapid heart rate. So symptoms include drowsiness, breathlessness, breathlessness, and a rapid heart rate. Okay? Now, these hydrogen ions, rather than lowering blood pH, instead they can join with hemoglobin, and I'm going to make that a bit longer because I'm not going to fit it on. They can join with hemoglobin to form hemoglobinic acid. So H plus joins with hemoglobin So the hydrogen ions join with hemoglobin to form the second molecule that we talked about earlier, hemoglobinic acid. Okay. So most cells in your body carry out aerobic respiration and release carbon dioxide into the blood as metabolic waste. Carbon dioxide is carried in the blood by carbaminohemoglobin and hemoglobinic acid in the blood to the lungs where it's exhaled out. So carbon dioxide, when it's released into your blood from your cells, can go one of two ways. First of all, it can just be joined with hemoglobin as it is to form carbaminohemoglobin. However, some carbon dioxide will be added to water via the enzyme carbonic anhydrase, which will form carbonic acid. Carbonic acid dissociates or breaks down to form hydrogen ions and bicarbonate ions. Bicarbonate ions just remain in your blood, okay? And then when your blood goes to the lungs, a different reaction takes place to get rid of it. Hydrogen ions, if they remain in the blood, it can lower your blood pH, which can lead to acidosis. Acidosis is an illness caused by low blood pH, and symptoms include drowsiness, breathlessness, and rapid heart rate. To counter this, what happens is the hydrogen ions inside your blood just join with hemoglobin to form hemoglobinic acid, so that it can be carried to your lungs where it can be gotten rid of or exhaled out. So that's carbon dioxide metabolism. Okay, I'm going to refresh the screen now, so if you have not got down all of the notes, you can pause the video and do so. We now need to think about nitrogen metabolism. Okay, nitrogen metabolism. So if we can put the subheading, nitrogen metabolism. And we can think about this process of deamination, something we spoke about on the first part of this lesson, and it is a key word. Deamination. Now, deamination is the removal of an amine group from amino acids in the liver. Okay? So deamination is defined as the removal of amine groups from amino acids or from excess amino acids in the liver. Okay? Now, what's important to understand is that the body can't store excess amino acids or proteins. Therefore, they have to be broken down in deamination. So I'm going to put in red underneath here because it's important that the body cannot store excess proteins or amino acids so they have to be broken down in deamination. Okay? Now, when this amine group is removed from an amino acid, it forms ammonia. Okay? So we can put underneath here again that amine groups removed from amino acids form ammonia. Form ammonia. Now, these are highly toxic and have to be converted into urea in something known as the Ornithine Cycle in the liver. So we can say that, in fact, I'm just going to carry on here, that ammonia is highly toxic so is turned into urea via the ornithine, ORNITHINE, the ornithine cycle in the liver. Okay, so deamination is the removal of amine groups from excess amino acids in the liver. The body can't store excess proteins and amino acids, so they have to be broken down in a process known as deamination. Amine groups are removed from amino acids to form ammonia. Ammonia is highly toxic, so it is turned into urea in the ornithine cycle in the liver. Okay, so if we're starting with proteins or amino acids, so we're going to start with proteins slash amino acids. It's not acid, it's acids. These go through what's known as deamination. So we can have deamination to form ammonia, which is NH3. Ammonia is toxic, so I'm going to put underneath here in brackets that it is toxic, and I'm going to put it in red so that it stands out. This then needs to be converted into what's known as urea in the ornithine cycle. And in the ornithine cycle, essentially what we're having is two ammonias, so two NH3s, being added to carbon dioxide, another waste product, to form urea, which is CO brackets NH2 brackets 2. And it also forms water. This is a summary of the ornithine cycle. Okay. Now, the urea, which is this part here, the urea is removed by the kidneys and stored as in the bladder as urine. So this urea is removed by the kidneys and stored in the bladder as urine. So proteins can't be stored. They can go through deamination, where they have an amine group removed from them. This forms ammonia, which is highly toxic. The ammonia can be added to carbon dioxide to form urea and water. Water is fine, we keep that in the blood, but the urea is filtered out of the blood by the kidneys and stored in the bladder as urine. However, Another process that can take place, which we haven't spoken about yet, but it's certainly worth mentioning because I'm sure you've heard lots about it online, is something called ketosis. So excess proteins can be turned into what are known as keto acids. Now, I'm sure you hear lots of influencers on social media and whatnot talking about keto, the ketogenic diet, and this is essentially where you turn proteins into other molecules. You turn them into things such as fats and carbohydrates for you to use as energy. So you eat a low carb diet so that your body turns proteins into carbohydrates and glucose so that you burn fat, but you also have energy to survive. Now, these keto acids from excess proteins and amino acids can be used in respiration, but they can also be converted into carbohydrates and fats. Now, when we make glucose and carbohydrates from another form of molecule other than carbohydrates, this is known as gluconeogenesis, which we've spoken about before. Gluconeogenesis. Okay. Now we just need to talk about the ornithine cycle. So we're going to focus on this part here, the ornithine cycle. I am going to refresh the screen, so you may wish to pause the video if you haven't got all of the notes down. So the ornithine cycle, which is part of the nitrogen metabolism. Ornithine cycle looks like this. So we have a molecule known as citrulline, which I'm going to put further down here. You'll see why in a moment. Citrulline being converted into arginine, which in turn is converted into ornithine, which in turn is converted back into citrulline. Okay? So to convert ornithine into citrulline, we're adding ammonia and carbon dioxide to make water. So we're going to add our ammonia and carbon dioxide to form a molecule of water. Okay? So we're also adding carbon dioxide. I'm going to put there CO2, add our arrow in. So we're adding ammonia and carbon dioxide to ornithine with removal of water to make citrulline. At the same time, ammonia can be added to citrulline to form again water to get rid of this ammonia, but it converts citrulline into arginine. Okay? And then to convert arginine into ornithine, we re-add some water and we make urea. In fact, I'm going to put it in red because that's what's gotten rid of. So water is added to arginine to form urea and ornithine. That is your ornithine cycle. It's not the neatest, I know, but essentially what we've got is ornithine being added to ammonia and carbon dioxide to form water and citrulline. The citrulline has ammonia added to it again, releasing water and arginine. Arginine has water added to it to form urea and ornithine. So it takes 2 molecules of ammonia to make 1 molecule of urea. So we can say that, what colour can I use? Use this orange. We can say that it takes 2 molecules of ammonia in the ornithine cycle to form 1 molecule of urea. Okay? There's essentially 2 condensation reactions and 1 hydrolysis reaction, which gives us a net gain of 1 water molecule. So we can say that there are 2 condensation reactions so we're removing water twice, and 1 hydrolysis reaction which gives us, or which gives a net gain of 1 water, or 1 water molecule. So we're removing 2 molecules of water, we're adding 1 molecule of water, which leaves us 1 left over. And what we're left with at the end of each turn of the cycle is 1 water molecule and 1 urea molecule, okay? So this breaks down 2 ammonia molecules into 1 urea, leaving 1 water left over. So we have ammonia and carbon dioxide added to ornithine, which creates water and citrulline. Citrulline is then added to more ammonia, which forms water and arginine, and then water is added back to arginine to create urea and ornithine. Okay? So that is nitrogen metabolism. As a final part of this lesson, we need to have a think about the functions of the liver. So I'm going to refresh our screen, send it back to black, and we can put the subheading Functions of the liver. And we can underline it. Now, to break the page up from my amazing drawings, I've prepared some images we can put in. Your liver essentially looks like this, okay? It's found in your abdomen, and it's this sort of long purpley organ. It's rather large. And essentially what the liver does is it makes bile, plasma proteins, it also makes cholesterol, and in the fetus, so in a developing baby inside of the mother's womb, it creates red blood cells. So, and red blood cells in the fetus. In adults, it doesn't make red blood cells because red blood cells are made in your bone marrow, okay? It also stores glycogen along with vitamins. So it stores glycogen and vitamins. Another job that it has is that it breaks down hormones and red blood cells. Breaks down hormones and red blood cells, or erythrocytes. And it also detoxifies the blood from alcohol and drugs. So, detoxifies the blood from alcohol and drugs. What else have we got? There is something else. And it controls blood glucose levels, okay? So, controls blood glucose, amino acid, and lipid levels. So just from this list, you can see that the liver has some really important jobs in your body. It makes molecules known as bile, plasma proteins, cholesterol, and it also makes red blood cells in the fetus. It stores glycogen and vitamins. It breaks down hormones and erythrocytes, which are known as red blood cells commonly. It detoxifies the blood from alcohol and drugs, and it controls blood glucose, amino acid, and lipid levels in the blood. Okay? Now, the liver is made up of what are known as lobules. But before we talk about that, we do need to write down how it breaks down ethanol. Okay? Sorry, I'm getting ahead of myself. So your liver can break down ethanol in your blood. Okay? Essentially, how it does this, so this is the drug in alcohol. For those of you that don't know. And ethanol in your blood or in your body is converted into ethanol by your liver. So it's converted into ethanol. It does this by removing hydrogen ions from it. So it is oxidized. The ethanol is oxidized into ethanol. So it's oxidized by removing 2 hydrogen from it. Now, in order to do this, our old friends NAD and NADH are used. So NAD accepts these hydrogens to form NADH. Ethanol is then oxidized again. So 2 more hydrogens are removed from it. So again, NAD accepts, that's going to annoy me because it's a different colour, isn't it? That'll drive me crazy. NAD is converted into NADH. The ethanol, when it's oxidized, is converted into something called ethanoic acid. Ethanoic acid. Now, ethanoic acid is converted into acetyl coenzyme A, again, a molecule we've heard about when we spoke about respiration. So it's converted into acetyl coenzyme A, which then is fed into the Krebs cycle. Okay, so this is showing how the liver detoxifies the blood of alcohol. So the liver uses NAD. So the liver uses NAD as a oxidizing agent because remember, oxidation is loss of hydrogen. The NAD is accepting the hydrogen from ethanol and from ethanal, so it is being oxidized. Therefore, NAD acts as an oxidizing agent. So the liver uses NAD as an oxidizing agent to break down ethanol in the blood. Okay, so the functions of the liver include it makes bile, plasma proteins, cholesterol, and it makes red blood cells in the fetus. It stores glycogen and vitamins. It breaks down hormones and erythrocytes. It detoxifies the blood from alcohol and drugs by using NAD as an oxidizing agent to oxidize ethanol into ethanal, into ethanoic acid, and then acetyl coenzyme A and the Krebs cycle. It also controls blood glucose levels, blood amino acid levels, and lipid levels. Okay, so final bit. I know I've said that before, but it's easier if we break it down like this. So we've spoken about the functions of the liver. We now need to talk about the structure of the liver. The structure of the liver. And we can underline that. Now, the liver is supplied with blood from what's known as the hepatic artery, which is here, and it also has the hepatic portal vein, which is, I can't see it here, the portal vein. Now, the hepatic artery brings oxygen from the aorta, whereas the portal vein brings blood from the gut, which contains lots of nutrients. So we can put this image in over here, again, to make it look a bit nicer. And we can say that the liver is supplied with blood from the hepatic artery, which supplies oxygen or brings oxygenated blood. So oxygenated blood or oxygen. And it also is supplied with blood from what's known as the hepatic portal vein. So it's supplied with blood from the hepatic artery and the hepatic portal vein. Which carries nutrients from the gut. So blood from the gut. Okay. Now, the liver is made up of what are known as lobules. So the liver is made up of lobules. Okay? Now, lobules contain lots of types of cells, and I've prepared an image taken directly from Google that looks like this. I'm going to make it a bit bigger so we can see. I'm going to try and fit it down here in this corner if I can. In fact, I'm going to leave it there like that. Now, lobules contain lots of cells. So, lobules contain various cells. The first one that we're going to talk about are these hepatocytes here. Hepatocytes. H-E-P-A-T-O-C-Y-T-E-S. Hepatocytes. Now, hepatocytes take substances they need from the blood to get rid of things like urea. So, hepatocytes, we can say, in fact, no, I'm going to word this different, apologies. They're involved in protein synthesis and making bile. So essentially, hepatocytes make bile and they make proteins. The hepatocytes make up about 80% of the liver. So they make up about 80% of the liver mass. So the majority of the liver is hepatocytes. Okay. Now hepatocytes do take substances from blood and release waste into the blood. So they take substances from the blood and release waste back out. Okay. Other ones include Kupffer cells, which are found here above the hepatocytes. So we've got Kupffer cells. Is it that colour? It is. It drives me crazy if the colours aren't the same. It's not the same, is it? Honestly, it'll send my OCD crazy. So Kupffer cells. Now, Kupffer cells break down old red blood cells. So these break down old or dead old erythrocytes or red blood cells, but we need to make sure we're using the correct term. They're essentially a form of phagocytic cells. So I'll put it in this colour. Phagocytic hepatocytic cells. So essentially what they do is they engulf old erythrocytes and break them down. We've got what are known as, and it's gone off the screen a little bit, cholangiocytes. Apologies, I can't really pronounce it. Now let me check the colours right. That looks same. So CHOLANG oocytes. Cholangiocytes. Apologies if I'm butchering that. If people are watching this video and they know that I'm saying that wrong, apologies. Now, these modify bile. So they modify bile. Essentially, the bile is made and these cells modify it. And finally, they're not cells, but we need to know about what's called a sinusoid. The liver doesn't have its own capillaries. These are replaced by gaps between cells known as sinusoids. So instead of capillaries, the liver has sinusoids. 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 And we can define these as irregular space irregular tubular spaces between cells for the passage of blood in the liver. For the passage of blood in the liver. So instead of having capillaries, the liver has sinusoids, which are these gaps between cells where blood passes through. So sinusoids replace capillaries. If I can fit that in, I'll be a legend. Replace capillaries. And lo and behold, I am a legend. Look at that. I know none of you are shocked. So the liver is supplied with blood from the hepatic artery, which carries oxygenated blood, and from the hepatic portal vein, which carries nutrient-rich blood. The liver is made up of lobules, and lobule is shown down here. In the middle, you would have the central vein, and around it you would have all of these Kupffer cells, hepatocytes, and this one that I can't pronounce, cholangiocytes. Hepatocytes are involved in protein synthesis and making bile. They make up 80% of the liver's mass, and they take substances out of the blood and release waste into the blood like any other cell in order to make these proteins to carry out metabolic processes. Kupffer cells break down dead erythrocytes, so Kupffer cells take out dead red blood cells and old red blood cells from the blood and break them down. They're a form of phagocytic cell. Cholangiocytes modify bile, which is produced by the hepatocytes, and instead of capillaries, the liver has sinusoids. Sinusoids are gaps between cells that allow blood to flow rather than having these capillaries. Okay? So essentially, this liver will be broken up into millions of these lobules, and blood vessels, particularly the portal vein and the hepatic artery, will feed into these. As it moves through, bile will be added. We'll get lots of removal of metabolic waste. We'll get deamination of excess proteins and all the other jobs that we've spoken about earlier. And all of the blood that gets to the middle of the lobule will be released through this central vein, which puts the blood back into the circulatory system. Now, in your specification, it asks that you are able to label a picture micrograph of the liver. So essentially what you need to be able to label is that you've got a central vein at the middle of a lobule. This is one lobule. From this central vein, we've got these pink cells coming out, which are hepatocytes. You've got all these hepatocytes in pink, sort of radiating around the central vein. And in between these hepatocytes, you've got these blank white spaces, which would act as sinusoids. The blood would flow through these, and hepatocytes would use whatever they need from the blood and add to it what they don't need. So, you should now be able to define the word excretion. You should be able to tell me the structure of the mammalian liver and explain its functions, and you should be able to evaluate stained images of the liver to identify key parts. I hope this lesson has been useful to you. If you have any questions at all, please get in contact with me via the usual means. If not, I will see you in the next lesson. Take care.